who are not here can understand what we're doing. Okay, so we were initially talking about the cytoskeleton, right? And then I told you guys that there are 3 of them that you need to know, and it was also your homework to go through what their individual functions are. So we talked about their structures. We know that there is a very small one known as a microfilament. We know that there's a very large one known as a microtubule, and between them we have an intermediate filament, right? And then we know that the intermediate filaments are made up of another structure, the microtubules of another structure, and microfilaments of another structure. These structures are modified proteins. We also know where the proteins are modified and packaged. We talked about those organelles. So briefly going over these again, and then this was your homework. So briefly going over these again, we have the, now just focus on the things my pointer goes to and do not read the extra information because this is a highly advanced diagram, but it's very essential for your knowledge. So this blue one here, these originating from the MTOC, and the MTOC, like I told you, were the microtubule organizing centers. And these excuse me, my battery is low. Can you give me a minute, please?
All right, Miss, sure.
Okay, coming back. So wherever you don't understand what I'm saying, just point it out. So very simple stuff. I told you the MTOCs were the centrioles. Initially, you guys told me that you thought these were the microtubules, to which I pointed out that these are not the microtubules because they're adjacent to the nucleus and it is in the form of a pair at right angles to each other, right? And then I also told you that you guys are right. The centrioles are in fact microtubules, but they're more specialized and they form the microtubule organizing centers, or the MTOCs, from which the microtubules originate. And that's essentially seen in cell division when we see that MTOCs are the exact points from which the microtubules help the cell divide. So then we know that the filaments originating from these MTOCs are definitely the microtubules. I hope you can appreciate this one, and this one right here, right? Going outside or inside, wherever, doing their functions. Now, I'm going to ask you what their functions are right after this. So I hope you've done your homework as well. But just going over it, I also told you guys that the microtubules are just essentially made up of 2 protein units, an alpha tubulin and a beta tubulin, joined together and to form a dimer. Look at this one and this one, they form a dimer. And rings of dimers sit on top of each other, crossing each other to form a tubule, along which things can transport, right? This is as much we did yesterday, and just a revision to make, because we also did Celia and flagella, and I just don't want you guys to forget that. Again, coming to the smallest, which are the actin filaments, they were just actin proteins, G-actin or any actin. You don't need to know the specific one, just know that they're made up of actins joined together, one chain and then another chain twisted upon each other. They've pointed out the actin filaments right here. So you can see the actin filaments right here. And then the third one are the intermediate filaments. As you can see, they're formed from units, and these units join together. And I also told you that these give the cell its shape. So before we move on, can you tell me which one function for each one of these, other than the ones that I've told you, or maybe including the ones that I've told you? So let's start with the smallest. What is the actin filament for in the cell? So,
Miss.
okay, you didn't. Did you read up on it, or did you not get time to do it?
Yeah, Miss. I'm sorry, I forgot.
That's fine. Okay, I had that slide prepared because that's fine. Honestly, So moving on, if we go on to the next picture. Okay, so very easy stuff. Again, going over the structures, we know that they're just actin beads. These are the actin beads forming single chains that twist upon each other. We know that intermediate filaments are just interconnected blocks. You can see one block here and many blocks joined together to form these. The substance inside them is different from actin and tubulin. It's essentially keratin or vimentin or lamin or many different stuff. So you don't need to know these, but in case they ever come up, you'll just have an idea of the words that are inside it, right? And then the microtubules, like I told you, alpha and beta tubulin. Okay, so just one thing. So when we say there's a positive end and a negative end to a microtubule, the positive end is the end that is towards the cell, and the negative end is the end that is going away from the cell. So that is one thing that you need to also know. But that's not, I don't think that's as essential in A level. So just to go over the functions. Now, the actin filaments are essential for cell shape. Okay, so they're essential for cell shape. What they basically do is that they help move organelles. So this is the cytoskeleton. Each one of them is going to give a cell shape. You can see the first line for each one of them is cell shape. That's what you need to know. And then essentially the only job of the intermediate filament is to hold the cell into its shape and hold the organelles into the position that they are in. So even if you don't want to remember this stuff because it's too much of written text, just know one thing. Intermediate filament give the proper shape of the cell, and they also hold the organelles into place. That's it. That's intermediate filaments for you. Moving on to the actin filaments. Now, the actin filaments are essentially also giving the cell its shape, just like we were talking about. Excuse me. Okay, sorry. We know the intermediate filaments are holding just the shape, but the actin filaments have more work than just holding its shape. They're useful in moving organelles from one place to another. They're useful in contraction of muscles. They're useful for the cell in either, you can say, crawling or moving. When will the cell crawl? When it's like an amoeba or something like that. Okay, so the actin filaments have multi-dimensional functions. Again, the microtubules are again used to hold the cell into a specific shape. You do not need to know this push and pull stuff. It's not at, it's a little more advanced than your level. But this diagram is very nice, which is why I chose to put it in here. But the important thing is you need to know the two examples of microtubules that we essentially talked about, the flagella and the cilia. If you remember, I told you guys, cilia are the organelles on the top of cell, for instance, in your nares and your nose passages, which move dust. So essentially, cilia are just extensions coming out of the cell that are thread-like tails. And then flagella is a very large tail that's essentially on a bacteria or amoeba that moves it. And what is it made up of? Again, microtubules, beta and alpha tubulin dimers, arranged in different shapes. Okay, so we also talked about the centrioles, and I told you guys that the centrioles are essential in cell division. So here it is, cell division. The specific thing in cell division is to divide the chromosomes. So you don't need to know the detail of this. It's going to come up in a later chapter, but just for completion's sake over here, we're putting in the word moving chromosomes separately within cell division, because as you will read later on, cell division has many parts. Okay, and then there's the formation of a cell plate. That's also in a later chapter, so you don't need to worry a lot about it right now. And then, like I told you, they're like basically railway tracks that move organelles on them. So if I ask you three independent functions, intermediate filaments anchor the organelles and the cell into its original shape. Write it down if you have to, but this is the basic function. Actin filaments, they help hold the cell into shape and move organelles. They can help move the cell as well. Microtubules form important structures like flagella or cilia, help move chromosomes apart in cell division, help move organelles as a railway track. So and the essential word here is intracellular transport. You don't see the word intracellular transport as importantly used here as it is anywhere else. So cell shape, intracellular transport, and this one is just cell movement. So I hope that that is clear.
Yeah, Miss.
So these three are the basic functions. You need to know them. Whenever somebody asks you, you don't need to learn them. Just know intermediate filaments. Okay, the middle ones. What are they supposed to do? Anchor organelles into place. Microtubules, the largest one. They have two. They make flagella and cilia. They make sure that there's intracellular transport taking place. And then actin filaments, they make sure that the cell is able to move and adapt its shape because they're very thin and flexible. They make sure that the cell can move and adapt its shape. Okay. Now, okay, going back to the slide that we talked about the cilia and the flagella. The cilia we talked about have a specific shape of its tail and a specific shape for its body. The tail, as we know, it has a 9+2 organization. We talked about that. If there is any question still, you can ask me. And then the body we know as it is has a 9+0 configuration. So here we have a little bit about microtubules right out of your book. It's so, as your book says, it moves secretory vesicles and other organelles. It helps in exocytosis, just like I told you. From Golgi vesicles, we get smaller vesicles that can get exocytosed out of the cell, right? And as we know, intercellular transport is a responsibility of microtubules because they are the railway system. So that's basically what's written here. We talked about nuclear division, that's written in your book. Like I told you, chapter 5, when we're going to do topic 5, we're going to discuss it in a lot of detail. Then we talked about the centrioles and how microtubules originate from them. And then we also talked about the cilia and the flagella. The flagella is essentially just a single long tail. It's very long and it's in some bacteria and animals, especially unicellular organisms, in which it helps with movement of the whole cell. Whereas the cilia does not move the whole cell. It's much smaller. It just helps move substances on top of the cell. Let me give you an example just to make it further clear. If this is an animal cell, these are many cilias. Originate tails of the cilia, this tail originating from here. Each of them is going to have a body down here, and these tails just essentially move like a dust particle or any particle along the way. Whereas a flagella is just essentially an outgrowing from either a unicellular organism or a multicellular organisms. It's a much longer outgrowing, and this helps move the entire organism down its path. So I hope that's clear.
Yes, Miss.
Okay, so moving on. Okay, so moving on, we talked about organelles called lysosomes, if you remember. Now, the important thing here about lysosomes that is additional information in A-levels is that there are two types of lysosomes, primary and secondary. Now, this is not wasteful information. Let me tell you, I still remember there being a question about the secondary lysosome, and unless you know this diagram, so this diagram essentially is not in your book, but in your book, there's one diagram in which they mention a secondary lysosome and a primary lysosome differently, which means they expect that you guys must know what a secondary and primary lysosomes are individually. So let me just give you a brief idea. If you remember from our original lectures, what apparatus is this? What organelle is this?
Golgi apparatus.
Yes, true, because it's not originating from the nucleus, but it's just a deposition of these tubules upon itself, and then there's vesicles budding out of it. So when a lysosome is essentially, like I told you, a sphere which has budded out from the Golgi apparatus, and it contains enzymes or certain substances in it. So when a lysosome combines with anything that we also talked about endocytosis in our last lecture, that substances, as they're being exocytosed, to balance that out, substances are being endocytosed. So imagine a food particle like glucose being endocytosed when this vesicle combines with a lysosome that is primary, which means it hasn't been used up yet. It's in its original form. It has budded from the Golgi apparatus. It is smaller and it contains inactive forms of the enzymes that are just made. And it has not combined with any food substances. So it has not underwent digestion. So it's not essentially performing any function as of yet. It just has the enzymes in their inactive original form. When this combines with with this vesicle that has been endocytosed with food, it's called a digestive vacuole. And when this digestive vacuole forms, this digestive vacuole digests everything in here. So this digestive vacuole is essentially a secondary lysosome. Now, if the food substances are coming from the outside, it's called a digestive vacuole. But sometimes we need to destroy old food substances or old organelles within our cell. For instance, an old mitochondria that is useless for us. So when these primary lysosomes combine with, now look at the mitochondria, because it's much larger in size, many lysosomes are attacking it. And when they combine together, they form a much larger secondary vacuole, a secondary lysosome. And this secondary lysosome in this case is an autophagic vacuole. Autophagy, as the word says, so phagy means eating and auto means self-eating. So autophagy, as the word says, essentially just means eating your own self. So if I hope this is clear, the primary lysosome, the original one, secondary lysosome, the one that combines and starts performing the function it's supposed to perform. It's basically combination of a primary lysosome and an endosome. The endosome can be substances from outside to form a digestive vacuole, or it can be an autophagic vacuole that is eating itself with substances from the inside. So you know what the secondary lysosome is. It obviously makes sense that it is much larger and it contains active form of the enzymes and it does digestion. And so what essentially happens is, now this lysosome can become, so the enzymes break down at many times because the substances inside will damage the enzymes to an extent. So essentially what happens is either the enzymes eat themselves up, see, because they're active. And we also know that enzymes themselves are also proteins, which mean they can destroy themselves. And then any extra content that is useless, for instance, let me say some water is left, some water is made up, that water can be exocytosed. So exocytosis is a process of the secondary lysosomes, not the primary ones. I hope this table is clear.
Yeah, Miss, it's clear.
It's very self-explanatory. If there's still any question, you can ask me. Okay, so now coming back to this diagram, the essential function of this whole lecture on topic 1 was to be able to understand this diagram. If you understand what each number represents, you're good to go. If you don't understand any of these numbers, ask me anytime during the lecture. Do I need to go over it again?
No, Miss, I got it.
Okay, perfect. Let's move on. There's a few remaining topics which are comparative topics. And so let me just tell you right here. Comparative topics tend to come up in 4 or 6 mark questions a lot. 6 mark questions don't come up as much nowadays. 4 marks questions do. So in comparative topics, you just need to know the difference between different cells. And since we've done the organelles, we can easily talk about the difference. So we know what's common in them. We know all of these. We know and reticulums must be present because we need food modification. From O levels, we remember that the plant cell has a large vacuole, whereas animal cells have smaller ones that store food. We know the Golgi apparatus, because we need protein modification, must be the same. Essentially, the different things in plant and animal cells are given right here in the bottom part of the image, which is a cell wall and chloroplasts, and the animal cell has centrioles. Now, that does not mean that the plant cell does not have microtubules. It does, but it does not have centrioles. Centrioles, like I told you, were essential in division of chromosomes in cell division, right? It does not mean that the plant cell does not divide. It does divide, just that the function that the centrioles performed here is performed separately in the plant cell. Again, we'll do that in chapter 5, so no need to worry about it right now. And the animal cell, again, has lysosomes, and that's literally as much as you need to know. You do not need to know centrosomes for now. And then there's one additional structure, a plasmodesmata. Plasmodesmata are holes between cell walls of different plant cells to allow movement of substances. As simple as that. Can you see, can you appreciate the hole over here in the cell wall? It's as simple as that. So this is again comparative, very easy. So you can go over it in your book as well. Not a difficult topic. We've been doing plant and animal cells thoroughly. When we're doing question papers, we'll also do it in detail again. Just for now, you need to appreciate the smaller stuff. For that, I have shown you an image from your book. This shows a plasmodesmata. Look at the cell membrane and cell wall apparatus right here. There's a hole to allow substances to easily pass through from one cell to another. Just need to appreciate this. Other than that, we've already talked about how there are grana in the chloroplast, how it has a double envelope, how the mitochondria looks like, how different structures look like, how the smooth endoplasmic reticulum is. Look, there's one smooth endoplasmic reticulum floating about here without ribosomes attached. A rough endoplasmic one floating about here with just around the nucleus with ribosomes attached. Here are the keywords. Very easy keywords. We've talked about what the plasmodesmata is. We've talked about the central vacuole. We haven't talked about a tonoplast, but if you remember from O-levels, tonoplast is the name of the membrane given to the vacuole. So this membrane right here, we don't call it a cell membrane. We specify it because the vacuole in a cell, in a plant cell, is larger and it has specific substances such as salts, sugars, and waste products. And this membrane surrounding it is simply known as tonoplast. Very easy stuff. We know what the grana are. They're stacks of membranes inside a chloroplast. Right here, like I told you, stacks of cells. Imagine that it's stacks of coins. They form one grana. And then there's one additional structure that you've done in O-levels. It's a microvilli. Microvilli essentially are just cytoplasmic extensions. They're very common in the small intestine. There's small cytoplasmic foldings or extensions inside the small intestine. And what is their function? To increase absorption. If you remember from O-levels, there were 3 specialized cells that you guys were taught in topic 1 of O-levels. It was a red blood cell, a root hair cell, and a microvilli. Can you appreciate and remember both of them?
Yeah, miss.
Okay, so if you remember from O-levels, you remember what these are. Please do not confuse the cilia with the microvilli. Microvilli are extensions of cell cytoplasm, essentially for absorptions or secretions, whereas what were the cilia? They were extensions of microtubules from the cell used for movement on substances on the top of the cell. And let me point it out to you here that if this is a cell and it's resting on a membrane over here, this area is known as the base. These are known as the walls, and this is known as the apex. So the microtubules, when I say they extend from the apex of the cell. Okay, moving on to the most important topic for today. Let's stick to my pointer again. So the most important topic is that essentially, when we say essentially, when evolution took place, there were 2 cells that formed. A very basic cell and then a relatively different cell. So essentially, we say that 2 cells were formed, right? Now, we use 2 words to define these cells. The first word is eukaryotic and the second one is prokaryotic. You know what pro means? It means first, original, one. And karyotic itself just means it comes from the word cell, imaging of the cell. So karyotic is just a cell. Now, there were two kinds of cells, a pro or original cell, or as the scientists thought so, and a eukaryotic cell, or a cell that is relatively, let's say, later discovered or more developed. So essentially, if you can categorize both of them, an easy way would be a eukaryotic cell is an animal cell. It's all the cells within us. And a prokaryotic cell is a bacterial cell. So you should be able to appreciate that a eukaryotic animal, not only animal, it can also be a plant cell, a fungal cell, any of these specialized cell, whereas the prokaryotic cell is just a bacteria, is the example is just a bacteria. Okay, so for now, let's just keep that in mind and move on to the topic. Okay, now essentially, let me just tell you the basic difference between a eukaryotic and a prokaryotic cell. You must remember from O-levels that these two cells had the difference of a nucleus in it. There's wordings around the line. One has nucleus, other does not. But then you always wonder, like what is the nucleus essentially? Is it the DNA? What is it? So let me just give you the basic definition that I feel like is the essential one to differentiate between both of them. A eukaryotic cell has internal membranes, whereas a prokaryotic cell never has internal membranes. It has no internal cell membranes. And what does that mean? We've talked about organelles that have internal membranes, organelles like the mitochondria, organelles like vacuoles, with single membrane, with a double membrane, Golgi apparatus, endoplasmic reticulums. Nucleus itself is just a bunch of membranes surrounding the DNA. So now when we're talking about the difference in a eu or a prokaryotic cell, the essential thing you should realize is the only difference that essentially exists is the fact that eukaryotic organelles can have membranes on the inside. A prokaryotic cannot. If you understand this, you understand rest of the topic really well. So we know since we're going about it, we can easily say, let's go over this table. This table says there is membrane-bound organelles in a eukaryotic cell, whereas this one does not contain a membrane. As simple as that. And so we can say that the eukaryotic cell does have a nucleus because the nucleus is membrane-bound. Does this structure have a nucleus? Nope, not at all. This DNA is floating about randomly without a covering of a membrane. So that then we can easily say that the prokaryotic cell does not have a nucleus. As simple as that. There are some examples given. For instance, like I told you, there is animal, plants, fungals. They're all eukaryotic cells because what it means they all have internal membranes, whereas prokaryotics have the example of bacteria. Archaea, you don't need to know about it right now. It's out of your syllabus, so don't need to know this one. Nucleus is present again, like I told you, absent here. So the size, because there are membranous organelles inside, the size is definitely larger. This one, the prokaryotic, has a much smaller size. And when we're talking about eukaryotic and prokaryotic cells, so we also know that the DNA replicates in cell division. The DNA replicates in the eukaryotic cell, whereas in a prokaryotic cell, what happens is basically the DNA replication in this one occurs. So if this is the DNA replication in a eukaryotic cell can occur at any point. It can start here, it can start here. But in a prokaryotic cell, the entire cell, if this is this DNA, will replicate as it is. It cannot start at one point. It's just supposed to occur and form an exact form of the DNA. It just starts over here and reads forward. So that's essentially it. We'll go into details of this in chapter 5 again. Eukaryotic cells can be multicellular because they have specialized networks within them, whereas prokaryotic are unicellular. If you keep a bacteria in mind, you can imagine how bacteria are always unicellular. So essentially, now when we talk about the DNA, since the DNA here is protected in a nucleus, it occurs in many chromosomes and many strands, whereas in a prokaryotic cell, we need the DNA to cluster together because it has no covering. If it's not clustered together, this DNA will run wild and get lost, which is why we have a singular loop of DNA coiled upon itself. So we have a linear loop of DNA right here. So there's also a difference in ribosomes. Ribosomes in eukaryotic cells are much larger because the cell itself is larger in size, whereas in prokaryotes are much smaller. If you need to know specifically, and if it ever comes up, the ribosomes in eukaryotic cells are 80S, where S is the Swenensberg sign, which shows where this essentially, it's just a sign that talks about how when in a test tube, if you put cells in a layer and spin them in a centrifuge, these cells of a eukaryote settle much lower, and this unit is known as a unit of S. They settle at a location at 80 on the test tube, whereas prokaryotic cells settle much higher on a location of 70. So these are known as 70S. And because, as you know, when you spin stuff, and if it's heavier, it will sink to the bottom, whereas if it's lighter, it will remain on top. Is that clear? Do you guys understand what the Swenensberg sign is?
Yep.
Okay, so it's very easy when you centrifuge something. If it's heavier, it's just gonna sink. So easy as that. Now we know prokaryotic cells, they do not have membranes, so no extra work is needed for cells to cross into the nucleus and come out. So the growth, the replication, making of proteins will be much, much faster. Organelles, like we talked about, are not present. Why are they absent? Because there is no cell membrane present to hold them into shape. Also, the hereditary information, both of them can store it, because as we know, we pass on hereditary information like color, height, the same way bacteria that have a certain property, for instance, let's say antibiotic resistance, can pass it on as well. So that's very simple. Now, a cell wall may be present in a eukaryotic and a prokaryotic cell. Now the eukaryotic cells are, if we say they're of 3 kinds or 4 kinds, the animal, plant, fungi, and protists, given right here. The animal one does not have a cell wall. Plants and fungi do. If you remember, plants have a cellulose cell wall. Fungi, on the other hand, have a chitinous cell wall, right? So that is a cell wall for us. The prokaryotes have a special cell wall, this green one right here, and this is made up of peptidoglycan. Peptidoglycan. Again, plasma membrane is present because obviously it holds the cell into its shape. Cytoplasm is obviously present. The only difference is appreciated here, can be appreciated in this table. They can also be appreciated right here, where I've shown you a cell wall. So this is within the bacteria. So this was the difference between a eukaryotic and a prokaryotic cell. And this one right here will tell you, it's from your book, and this is gonna give you structures of a specific prokaryotic cell. Stay with my pointer right here. This specific prokaryotic cell is a bacteria. And this bacteria has some structures. For instance, it's always going to have a cell membrane. It's always going to have a cytoplasm. It's always going to have a circular DNA, just like we talked about. A DNA coiled upon itself, it's connected throughout, it's circular, there are no loose strands because there's no nuclear covering. We need it to be a circular DNA. It always has ribosomes. What ribosomes are those? Those ribosomes are 70S ribosomes. It will always have a cell wall. And what is that cell wall made up of? peptidoglycan. A peptide and a glycan. Glycan, like we talked about, glycans are carbs or sugars, peptides are proteins. So it has a protein sugar cell wall. It's also called a murine cell wall. Alternatively, we talked about a flagella. We said it's just a microtubule. Look at this one. Can you appreciate this? Some bacterial cells or prokaryotes may have a flagella. Those organisms will be called motile organisms. Some do not. Those will be called non-motile. You can appreciate that the microtubule tail is here and there's a microtubule body right here. Can you see this dot connecting to this tail? The body. Some of the prokaryotes may have a slimy capsule on top of them to further protect them, because obviously there's singular cells. They're not multicellular like us. They need super protective conditions inside whether they're inside the human body or outside. Okay. And then another extensive structure, one extensive structure from the cell is a flagella. We know what that is. Another one is a pilus, or a group of them called a pili. They're also extending from the cell. These extensions are known as the sexual organelles of the bacteria. For instance, if this is a bacteria and this is another bacteria, this bacteria has extensions from it called pili, and this one has extensions called pili as well. When these extensions meet, they form a sexual communication between each other called conjugation. Now they can sexually replicate. Otherwise, bacteria, like I told you, just replicate to form entire forms of themselves. They replicate basically horizontally. Horizontal replication, if you remember from O-level, just means that it's going to divide by binary fission into two of these molecules of bacteria, whereas vertical one means there's one cell is going to go down to form two different cells. If this is sexually, then another cell combine with it to form one complete cell with complete pair of chromosomes. So sexual transmission occurs here, and we know that in sexual transformation, half of this cell's bacteria DNA will enter in this cell, and half of this cell's DNA will enter in this cell. And this is going to enter through organelles called pili right here. These pili can also help cells to attach to each other or to different surfaces, and they're essentially for sexual reproduction. Other organelles within the prokaryote, there's an organelle called a plasmid. This plasmid is essentially just a circle of DNA. Do not confuse this. Do not think of this as a cell membrane. Do not think of this as a lysosome or a vesicle. It looks cellular, but look at the color. This color and this color is similar, which means this is the same stuff. So this is just a DNA, and because DNA is always double-stranded, so it's just two strands of DNA forming a plasmid like this. This DNA, the function, the specific, so we know the original DNA of the cell is always circular. So what's this DNA? This DNA is the form that can come in and attach to the normal DNA of the cell, or bud off from this cell's DNA to go through the pili into another cell. And essentially, when we talk about antibiotic resistances, or a non-pathogenic strain becoming a pathogenic strain, this is what ends up happening. DNA from this will detach and form a plasmid. The plasmid will then enter into another cell that, let's say, was essentially non-pathogenic. It was not disease-causing, and it will now become disease-causing. So I hope that is clear. Do you understand what the plasmid does?
Yeah, miss.
So it's just a small circular piece of DNA. And it's not the main chromosome. The main chromosome is this circular DNA. In animal cells or in eukaryotes, it's the DNA in those is called a linear DNA. In here, it's called a circular DNA. What it does is it provides extra genes. These extra genes can provide resistance to antibiotics. They can make non-pathogenic strains into pathogenic ones. Like, for instance, if there's a bacteria in the soil that causes diseases in plants, now that bacteria transfers its plasmids into a bacteria that causes diseases in animals. Now that new bacteria will cause diseases in both plant and animal, right? So that's what a plasmid does. And then and then the last thing is that the cells have, so this, the last thing is called a mesosome. This is a mesosome. It's essentially outgrowing. So because we know that the cell does not have internal membranes, what happens is the cell membrane itself can fold onto itself to form specialized outgrowings called mesosomes. And now these mesosomes can perform specific functions, for example, photosynthesis, nitrogen fixation. If you've done nitrogen cycles in chemistry, you'll know what that is. It just fixes nitrogen from the air into the soil, and oppositely. And so essentially mesosomes can be these, or mesosomes can also be, for instance, if I was making a sample of a bacteria and the cell, I damage the cell membrane and the cell membrane folds upon itself, it can form an internal folding called a mesosome. And so it can just be damage to the cell membrane, or it can be functional for the cell. So it depends on different functions. Just know that this infolding is called a mesosome. Okay, and then we've talked about the peptidoglycan, that it is made up of amino acids and a polysaccharide or a glucose chain combined together. And it can also be called a murine. So I hope prokaryotes are also clear.
Yeah, miss.
Any questions up till now?
No.
No questions. Okay, so then we can move on to what we call the most important stuff, which is the most new stuff in this topic, an electron microscope, looking through an electron microscope. So when we're looking through an electron microscope, we see images that are different from a light microscope, which is why I find it essential to talk about these images so that you can appreciate them in case they come up on your paper 3s, paper 1s, or paper 2s, anywhere. So this is a cell through an electron microscope. Like I told you, to obtain a flat image, we use an electron microscope called a transmission electron microscope, right? Can you appreciate the nucleus?
Yeah, miss.
The larger nucleus. Can you appreciate these fine little strands of endoplasmic reticulum? You can appreciate the nuclear membrane. You can appreciate some mitochondria. I understand this image is not as clear in resolution, so we have more coming up, and you can also appreciate the cytoplasmic membrane right here. So just have an idea, a black and white transmission electron microscope of any cell. Look at this one. This one comes from your book. Can you appreciate the nucleus?
Yup.
Within the nucleus, can you appreciate this cluster body called the nucleolus? Look at here, there's a double layer nuclear envelope. Can you appreciate the double layer? This is how sensitive electron microscopes are. Can you tell me why this is colored? I told you it's a black and white image. How do we get this colored? Because this one in your book is colored. Let me tell you, this is colored because we insert these colors within the computer. It does not come from the microscope itself. It comes from the computer. Can you appreciate to me an endoplasmic reticulum extends right after the cell nuclear envelope? Here it is, endoplasmic reticulum. Can you appreciate it?
Right, miss. Yeah.
Yeah, look at this. Look at this one here again, endoplasmic reticulum. There's some over here as well. And then what's a Golgi apparatus? Resembles the endoplasmic reticulum, but it's a little bit further away, and it has specific shape of its face, a cis face shaped like a cave or concave, and a trans face, right?
Oh.
Yes, can you appreciate it now?
Yeah.
Look over here. Again, we have a cis face maturing here, Again, a cis phase maturing over here as well, and very small trans phases. Maybe these are two different Golgi apparatus. They're making specific proteins, and both of these proteins need to be combined together here before being released together.
All right, miss.
Do you get the concept now?
Yep.
Another cis phase right here, another trans phase right here. And then also, let's also appreciate let's also appreciate that we have mitochondria right here. Can you appreciate this mitochondria? And it has a light blue line inside.
Oh,
This
yeah.
light blue line shows the cristae, the internal foldings. Not very clear, but you need to take some time to appreciate it. And this cell has a lot of mitochondria, a lot of this blue stuff, which means it's carrying out a lot of ATP synthesis.
Miss,
It's a
how about the
very active cell,
centrioles?
right? Can you appreciate that?
Yeah, miss.
And then let's also appreciate a cell membrane. Okay, let me just teach you how to do that. So you see some gap over here, right? There's some gap over here. And then there's some gap over here, which shows that there is something occurring over here, right? Now, either this is an entire cell over here, or this is two different cells. And you can see this thin yellow line separating them. So this means that this is one cell membrane, this is one cell, and this is the other cell. Can you appreciate it?
Oh, I see. Yeah.
Yeah, can you appreciate the cell membrane over here? Let me show you. Look at this cell membrane over here. And this is another cell as well, because you can see the intracellular space over here. Intracellular space over here as well. And look at these extensions of the cell, these tiny extensions. These are the microvilli. Can you appreciate them?
Yep.
Look at this extension right here. And this is essentially, where do we have microvilli? In the small intestine. If you remember from all of us, we have microvilli in the small intestine. And these
for the secretion
and they help in
absorption.
absorption, right?
Yep.
And because so much stuff is being absorbed over here, so much ATP is being made, which is why we have a lot of these blue mitochondria. A normal cell is not going to have as many. Look at this normal cell. If I say this is the mitochondria in a normal cell, this has very few, right? But this one is loaded with mitochondria. Why? Because small villi indicate that this is a cell from the small intestine. I hope this is clear. I understand it's a bit confusing and overwhelming, but I hope this is clear.
Yeah, miss, it's clear. I'm getting it.
Okay, perfect. And then you can also appreciate these green ones, vacuoles, just spheres with substances in them.
How about centrioles? Are they?
So you can't essentially see centrioles. Can you appreciate any cytoskeleton in this?
Nope.
That means if centrioles are part of the microtubules, you cannot appreciate them, right?
Ah, I see. Yeah.
Yeah, so now you know how to differentiate. You just have to put in a little bit of logic. That's it, nothing more.
All right, miss.
Okay, just to go over it, do you see this is like a very zoomed in image? What is this organelle? Give me a guess. Give me a wise guess. Think about it. What is one organelle that has an external membrane that is flat, but an internal membrane that is highly folded into cristae?
Oh, yeah, mitochondria.
Yeah, that's it. That's the mitochondria.
Miss, I was gonna say, but then it looked so different here, and I'm like,
well, because I feel like this is your first time going through electron microscope images, so I understand it's a bit daunting.
Yeah.
But like, if you understand these, you're going well. This was very small over here. Then we did it. We zoomed in into the electron microscope. Do you see the power of the electron microscope now? It even shows you those ribosomes. Look at these red dots. Yeah, red dots weren't visible in the previous slide because it wasn't as zoomed in. But look at them over here. And then I told you, if you can see two lines apart, if you can see this line and this line apart, it's called a good resolution. And good resolutions are a part of electron microscopy, right?
Yeah.
Just like good microscope zoom ins and good zoom in powers, good resolutions are also part. Okay, look at this cell again. We have a mitochondria. How do we call it a mitochondria? Because we can see this internal cristae, one line of this cristae, but this image isn't as good as its quality is supposed to be, but that's fine, I hope. Look at the nucleus. There's a nucleolus inside, a dense folding within the nucleus. Look at the double membrane of the nucleus, the nuclear envelope. Can you appreciate the double membrane?
Yeah.
And then what are these? What's this?
The rough endo.
Yes, it is the rough endoplasmic reticulum. You can't appreciate the ribosomes as well, but you can say it's the endoplasmic reticulum, right? Because it's extending from the outer membrane, and then it has a lumen in it, and you can separately see the outer lines, right?
Yep.
Do you get the diagrams? Do you get the diagrams now?
Yeah, miss.
So let's go over a plant cell without reading the labels. Let's see this cell. We know that it has a central vacuole. We can appreciate that here. There's supposed to be a nucleus. The largest membrane-bound organelle is a nucleus. If this is the nucleus, can you appreciate the boundings of this nucleus?
Yep.
Relatively darkly stained as compared to the rest of the cytoplasm. What's this folding? What's this condensation inside the nucleus?
Nucleolus.
Perfect, nucleolus. And then you can also appreciate that if these are, look at this granum formation. Can you see that there's, we have granum formation over here? It's like resting on top of each other. Even if you can't see it, you do know that the other largest organelle comparative to the nucleus is only one in a cell, in a plant cell, but the largest that are in many numbers and are membrane bound, double membrane bound, are the chloroplasts, right?
Yep.
So this makes it the chloroplast. Very easy. You don't need to cram the diagram, just need to know how each one looks like, and you can easily tell. So you're not going to go back and cram the diagrams. You're going to go back, make sense out of them, and then label them. As easy as that. This is to appreciate the double line, the good resolution of the cell membrane. Can you appreciate a bilayer, one layer and a second layer?
Yep.
So if somebody shows you this image, could you say that it's a cell membrane?
Yeah, miss.
Because it has a bilayer, right? Okay, so which electron microscope is this one? We talked about two electron microscopes.
Is this the scanning?
Yes, a 3D image.
Yep.
You guys asked me yesterday the difference between a scanning and a transmission one. Look at this one, a flat image, a detailed flat image.
Yeah.
And this is the last topic for today as well. A virus. And what does a virus look like? A virus is acellular, it's dead. It's just one cell that is dead. If it comes inside either a prokaryote or a eukaryote. So it's not prokaryotic or eukaryotic. It's just a virus. It's on itself, a separate class, a separate species. It can only become active when it affects a pro or a eukaryote, which means it can affect both a bacteria and animal, plant, fungi, right? And the structure is the easiest. It has a genetic code that, like all organisms, that genetic code can either be DNA or RNA. It is surrounded, so it does not have a membrane, it does not have a cell membrane, it has a capsid. Dead material, made out of dead material, a capsid, mostly proteins and some fats, etc., A capsid. So the essential structure of every virus is genetic material. Genetic material is the essential structure of everything, right? And in plants, we know that plants, animals, eukaryotes, we know it's linear genetic material in a nucleus. In prokaryotes, such as bacteria, it's circular genetic material, not in a nucleus. And both of them are DNA, but in a virus, it can also be RNA. Please appreciate that it can be either DNA or RNA. That is one difference between viruses and the other two cells. The second difference, it does not have a cell membrane. It rather has a capsid that covers the DNA or RNA. And then it can have an envelope proteins for greater protection. As simple as that. Appreciate this. Look at this envelope. Look at these extensions. They're envelope proteins. When we talk about the COVID virus, it's just an envelope, and these spike proteins, the famous spikes, what are those? They're envelope proteins, right? And on the inside, all it's going to have is either an RNA or a DNA surrounded by a capsid. As easy as that. Very easy structure of a virus. So essentially over here, what they're showing in this electron microscope is that if this is a cell and these are organelles within a cell, then a virus is as small as an organelle. It's this small. This black block is the Zika virus. If this is the Zika virus, it's inside that cell, right? It's as small and it's infecting the cell by being inside it. So how does a virus replicate? How does a virus survive? So we know it's acellular, that it's dead, because it has nothing else. It has no mitochondria, no ATP making things, nothing. The virus, how it survives is that it has enzymes here. This is an enzyme. This is an enzyme. What happens is when it infects this cell, for instance, this bottom of my screen is a large cell. This virus will help by these proteins. It's going to adhere to the cell. And these proteins and the envelope will then make a hole in this cell and the virus will enter. Once this virus enters into this cell, it will unfold and release its genetic material right here. For instance, if this is RNA material, the RNA material comes here. The enzymes are also released. The enzymes will then help replicate this RNA material into either a DNA or further RNAs that will make new proteins. And these proteins will then go into the nucleus of the cell or remain outside and hijack the material of the cell to make more and more and more and more viruses. And when this cell is filled with viruses, it has so many viruses that this cell will burst. And these viruses will be released to infect nearby cells. Is that clear?
Yep, miss.
So you understand how a virus lives and dies within a cell and takes it down with it.
Yes.
Perfect. Okay, so I think that much is it for today. There's a lot to absorb.
Yeah, miss.
Okay, we have another student who's here as well. Don't worry, I have a recording. I've recorded this lecture for you guys.